Good morning everyone, myself Lokeshwani and today I am here to teach you all science. In science today basically we are going to learn about cells, the building block of life. See, before I actually start with my respective topic, I would first like to tell that I have changed my teaching method now. Why? Because as you all know, CBSE has launched new NCERT book known as Exploration, and I think they have taken quite good decision because in this new NCERT many questions are also given in between, competency based questions are also there, and many more things. Fine. So what I was saying, in this books many colorful diagrams are also there, and for me it was not possible to show you all these diagrams in Google Docs from where I make my notes. So I decided why not to study line by line from NCERT, so that even you will read the NCERT and you can also get the answers, and you will also see the diagrams and will be able to relate with them. Fine. So let's start. The first para tells us that life is originated in water. Some researchers also believe that life may have originated in small water pools with changing environmental conditions rather than in the oceans. Now, this line means that, according to some scientists, it is understood or they think that life originated in water. Why? They specifically thought that it originated in some small water pools with changing environmental conditions rather than the oceans. Fine. So life originated in water, but specifically it originated in small water pools. Why only small water pools? Not in oceans. Oceans too is so wide. Why not there? See, I am telling you why it did not originate there. Because in water pools, environment changes faster. The environmental conditions change faster than oceans. Fine. Hot springs are examples of such environments. Such environments means the pond from where life is originated. Hmm. In India, the hot spring of Puga Valley in Ladakh mountain is very high, maintain very high temperature, nearly at the boiling point of water. What is the boiling point of water? 100 degrees Celsius. Even in a cold climate, these environmental conditions seem to be similar to those on the early Earth about 3.5 billion years ago. Why? Be- Because that time also life originated in water, and this time also. So there, because of all these, you can say water hot springs, the temperature remains same as it was 3.5 billion years ago. The organisms living in these hot springs are mostly heat-loving bacteria called thermophiles, which are unicellular. See, as I told you in starting only, that in scientific community they believe that life originated in water. Fine. So the specifically in hot pond, hot springs. Why? Because their climatic conditions change faster rather in ocean. So the bacteria which live in these hot springs are heat loving. So that's why they are known as thermophiles. What they are known as? Thermophiles. This term please highlight, that they are known as thermophiles, and they are unicellular. Unicellular means made of one cell, and multicellular means made of many cells. And we'll solve this, think it over also. Fine. After reading this whole page. Fine. And we'll also be practicing the back exercises, because there's so many good competency based questions are given. Fine. Up, scientists from Birbal Sahani Institute of Paleo-Science, fine, Lucknow, studied these Paleo-Sciences, Lucknow studied that these hot springs are found in that calcium carbonate. Now, read it properly. From here, MCQ type questions can come, and it is one of the best reasons that I decided to make you study from NCERT, so that I will read each and everything. See, notes what happened, each and writing each and every line is not possible. But here I am teaching you line by line, so that I can even tell you the NCERT, the MCQs and all. Fine. Scientists from Birbal Sahani Institute of Paleosciences, Lucknow, believed or studied that hot springs are found that calcium carbonate formed rapidly around them. Fine, देखो, the hot spring, hot spring, bubbling hot spring. So around the see, around the hot spring, this calcium carbonate get formed rapidly. These deposits may have protected early organic molecules from harmful radiation and extreme conditions, and they may have also helped in the formation of the first protective membranes, the barrier that defines the cell. Now, देखो कुछ इसमें टफ नहीं है. It just says that the scientists from Birbal Sahani Institute of Paleoscience, Lucknow, have studied that in hot spring, calcium carbonate gets deposited on the border. So they have related this scenario with the earlier time, that in earlier time also this calcium carbonate used to get deposited, and thus it used to protect the organic molecules, and when the organic molecules used to be protected, they used to form first protective membranes, which is the barrier that defines cell. Fine. Question can come, like they can ask you the name of the institute or the paleosciences name, and they can ask you what used to get deposited, and they can even ask you that give reason hot spring are and found. They can ask you that it is found that hot spring, they can ask you like this, that it is found that calcium carbonate is formed rapidly around hot springs. Why? So you will write whatever I have told you. Now let's come on the next topic. It says that all living organisms are made up of cell. Fine. You know that all living organisms are made up of cell. Suppose you are building your house. Fine. You are building your house. So the main raw material that you require to build your house is what? You require bricks. Fine. In same ways, for building an organ system, the main, you can say, base is cell. What is it? Cell. When cells group, they form tissues. If you know about tissues, very good. If you don't know, whenever it will come in your syllabus, I'll teach you. Cell, tissues, then tissues group to form organ, and organs group to form whole organ system or human body. Organ system. Fine. Now, let's read that all living organisms are made up of cells. The cell represents the basic level at which life exists. I told you that cell is the basic level which represents the basic level at which life exists. Some organisms such as bacteria or yeast consist only one cell. Fine. One cell means they are known as unicellular, while others, plant, fish, birds or human, are made up of millions of cells which work together. So they are known as multicellular. Fine. Made up of single kind of cell, unicellular. Made up of many type of cell, multicellular. Now, where does a cell come from? You have to think it over. You have to think from where does a cell come from? How have technological interventions facilitated the creation? These are the questions on which you should think. Okay. So, whenever you are trying to do this question, the first word that should come is why. Why? Okay, whenever you're studying from cell, you need to get understand from where does cell come from, how does cell multiply, how is the cell structure and function unit of life. So we learned about these all in our this video. Fine, let's move on our next topic. Up here, one line is that a group of similar cells performing similar function form tissues. Means a group of specific cells which perform specific functions. They are known as tissue. Different tissues are organized to form organ. See, whatever is written in this para, I explained you with help of an example. Here it is written different tissues are organized to form an organ and several organs work together to form organ system. I explained you here. Fine. Such as nasal pores, nasal cavity, trachea and lungs form respiratory system. You know what is respiratory system? How does respiration work? When oxygen reacts with when the food taken carbohydrate reacts with oxygen, it produces carbohydrate energy, etc. Fine. Up. Even when cells are organized into tissues, organ and organ system, the cell remains the fundamental unit of structure and function in all living organisms. See, this line tells us that, suppose I will tell you this the whole I will explain you this whole line with help of one relevant example. Suppose you built your house. Fine. आपने अपना हाउस बनाया. First you built of one floor, then the house was built of second, then of third. Fine. The building block was bricks in the first floor. When you build a second floor, so did it change? Did you build a second floor of plastic? No. Again it was brick. Third floor was also brick, and so on. The brick will remain same. So how can you relate this example with that line which I read? See, here it is. Here it says that when cells are organized into tissues, organ and organ system, means when cells from organ system or tissues, the base which is tissue, it remains the same. Base is not going to change, and the base which is cell, it functions in all living organisms. This makes us wonder that how such tiny cells perform so many different activities. Yes, absolutely. How such tiny cells perform so many different activities. What are the different components of a cell? How do cells in our body communicate with each other? Do cells live forever or they die? In this chapter, we'll explore about the answers to these questions as we enter into the fascinating world of science. Cell. First question is how to study cell. Now, here one diagram is given. This diagram tells us about the size of cells. Fine. Here conversion is given from which you can get to know about the size of the cells. First is small molecules, then atom. Nm represents nanometer. Here the sizes are given like protein, lipids, small molecule, atom. How big are they? Fine. Now we are coming on the topic which is how to study cell. What do we call the ability of the human eye to see two very close objects as separate and distinct? Means suppose two dots are drawn on a piece of paper. Now imagine this is a piece of paper. Two dots are drawn. Fine. As the dots are moved closer, there comes a point at which they can not longer be seen as separate. Dekho, you can try this activity right now. That take a paper, make two dots, bring it very close to your eyes. and one point will come that they will not be seen separately, only one point will be seen. Fine. So as the dots are moved closer, there comes a point at which they can no longer be seen as separate. When viewed about 25 centimeter, 25 centimeter means, suppose here is your eye. Oh, I'm not that good at drawing. Here is your eye, and means 25 centimeter away you have kept the paper. Fine. 25 centimeter away. The near point of the human eye. 25 centimeters is nearest point of human eye. Fine. Nearest point of human eye. Point of human eye. 2 points separated by about 0.1 mm can be seen as distinct. Otherwise, they appear as single point. This is called the limit of resolution of the human eye. What is the a question can come in your examinations, define limit of resolution. So it is nothing. You will just write that see, here specific definition is not given, but if you want to write, see, first understand it properly. That suppose you took a paper, drawn two dots, you brought it closer about 25 centimeter. 2 points are separated by about 0.1 mm can be seen as distinct. Fine. 25 centimeter they can be seen as distinct, otherwise they appear a single point, and this is called a limit of resolution of the human eye, which is 0.1 mm. Which is 0.1 mm means distance between these 2 points, 0.1 mm. A cell is usually too small to be seen by unaided means naked eyes or from your eyes. This raises an important question. Now one more question is there, that how have cell biologists studied the structure and function of the cells are much smaller than the limit of resolution of the human eye. Limit of resolution of the human eye is how much? 0.1 mm, and they are much smaller than that. So how have they studied? Now this question is there of physics from class 8th, chapter 10. Fine. You better know what that chapter 10 was. You have learnt about convex lens. Fine. You have learnt about convex lens. It is like this, the outermost, this convex. Fine. You know how does it reflect light? The surface which is bulging out, it reflects the light. Something. Fine. Wait, let me draw it perfectly. How does it reflect light? See, there are many conditions for drawing. So whenever any condition come, according to that we will be drawing. A convex lens or a combination of lenses, that is an objective lens of an eyepiece, are used for the magnification of an object to make it appear larger. Fine. So convex, why is it used for magnification? You know what is magnification? The process of making an object look or appear larger. So why? Because the bulge, as I told right now also, that if you see it properly, convex mirror is this, the part which is bulging out, so it reflects the light. That's why it is used for magnification, and when it reflects the light back, the object appears larger. Fine. Robert Hooke was the first ever person to observe a cell and see that cell was a dead cell. Robert Hooke was the first person to observe cell in 1665 using a self-designed microscope. Self-designed microscope means he had designed that microscope on his own, and under that microscope he studied. What did he study? He studied a thin slice of cork. Cork, you know, Cork and epidermis. Have you read about cork? Are the dead cells arranged compactly in the plant. They mostly from the bark of the tree. Bark of the tree. Fine. So he studied that cork under microscope. So he found out that there are some box like compartments. He named them cell. In school laboratories, light microscopes are used to observe. Right now, okay, fine. Now we are talking about in schools, physics laboratory. No, no. For microscope, though it is in biology lab. Okay, biology. In biology laboratories, light microscopes are used to observe objects using different objective lenses. Now, there is one diagram. Diagram is nothing, just structure of light microscope is given. Now, eyepiece, here you can adjust your eyes. Coarse adjustment, this knob is used to adjust the body tube. Objective lens focuses on the object which is played. Stage is where the object is placed. Mirror, the base for keeping it still. Handle is given and fine adjustment knob. See, some might wonder that why is mirror given here. So some main function is not there. It just acts as a light reflector. It reflects the light so that the person who is watching the object can see it more clearly. And next comes our fine adjustment knob, which is used to adjust. Now, different objective lenses. See, lenses example 10x or 40x, they are used to achieve better magnification and resolution under visible light. Explode now. When activity is given for you to explore the different parts of microscope in the school laboratory and use it to observe fine structure. Whenever if your teacher takes you to lab, so please go and observe. Under the microscope, you will see magnified image of an object. See, you will see the magnified image of an object. Can you estimate its actual size? So for that process there, but one formula is there, that is image upon drawing. Fine, but for that many steps are to be followed. Let us estimate the size of a cell. Now you will be estimating the size of a cell. Take a transparent ruler of millimeter. Millimeter, you know, it is in millimeter only. Place the ruler on the stage of microscope. Stage means here. Here you will place that ruler. Fine. Focus on it using the adjustment knob. Adjustment knob is where here. With this adjustment knob, you can focus more on the object. Fine. And observe the diameter of the circular field. Diameter, you know, a chord passing like this. Fine. This is the diameter. Through the eyepiece and measure it in millimeter. Fine. Observe the diameter of circular field of view. Circular field, whatever it is coming, that. And now convert the diameter from millimeter to micrometer. Micrometer की unit is this. See. Fine. Suppose the diameter of the visible field is 5 millimeter, and see, for converting it into micrometer, you just need to multiply it to 5. If it is coming 5 millimeter, then for converting it into micrometer, you need to multiply it with 500. Then don't forget to put the unit. In your examinations, this can come that if the diameter of the visible field is 5, let's say 7. So what is it in? What is the value in micrometer? So some people might write 7,000, which is correct, but you need to write the value as well. Without showing the value, no marks will be given. Fine. Remove the ruler and place onion peel slide on the stage. Slide means you will take one clean mirror type. You will place the onion peel. Now you will focus on the side, on the slide. Slide, you know, on a transparent thin like this, on which you have kept the peel. Fine. Now focus on the slide and count the number of cells present along the diameter. Diameter, present along the diameter in a straight line, and estimate the real size of the cell using this formula. How will you? 1 millimeter equals to 1000 micrometer. And for if you need to find out the estimated size of the onion peel, so diameter of the visible field upon number of cell along the diameter. First you found out the, if you need to find out the diameter, or no sorry, yes, estimated size of onion peel, तो first you will do that जितना भी diameter की side आ रही है upon number of, and this should be micrometer. Fine. Upon number of cells using the diameter. Now suppose 25 cells are seen along the diameter. In this case onion cell would be, अब देखो 25 cells are seen, and what is the formula? Diameter of the visible field in micrometer upon number of cell. So what is the diameter of the visible field in micrometer? 5000 upon 25. Yes, will go 200 times. 200 is the 200 micrometer is the answer. Don't forget to put the unit. Fine. Now we are coming on the next topic, that is structure of the cell. Now whatever we have discussed, I want you to revise. The next rest over topics we'll be discussing it in next video. I don't want for you people to make it hectic. We'll cover it slowly, but we'll go in detail and cover everything. Fine. If you have not, I think if you have not understood this part, estimated size of the onion peel cell, I can explain. You can see. For that you need to keep the ruler, focus on the rectangular circular side, and whatever the diameter is coming, find diameter of the visible field. Now on a clean slide you need to keep onion peel. Now focus on the microscope, focus with the help of microscope, whatever cells are coming the diameter you need to count. Fine. Now whatever the diameter was of the whole circular region upon whatever you have diameter you have got, whatever diameter of the cells you have got, number of cells in a diameter. And see, when you will focus in the rectangle circular region, you will get the answer in millimeter, but don't forget to convert it into micrometer by multiplying by 1000. Fine. So suppose you got 5 millimeter. I am explaining you what you need to do. You need to take a ruler, on that you need to focus on the circular region, whatever the diameter of the coming, or whatever the diameter of the circular region is coming, that should come in millimeter. And for now with the help of, by taking millimeter as a basic unit, we can't do the calculation. So for that you need to convert it into micrometer, and you for converting it into micrometer you need to multiply it by 1000. Fine. So diameter whatever you have got, that you need to convert into micrometer. Suppose you got 5 millimeter diameter, you will convert into micrometer, that will become 5000 micrometer. Don't forget to put the unit. Now what you need to do, you need to take a clean slide, place onion peel on that, and after that you need to observe it under the slide. Now whatever the number of cells are there in the diameter में जितनी भी cells हैं, you will write a number. Now for finding out the estimated size of cell, you will place you will write a diameter, what was the diameter, 5000 micrometer upon 25, answer will come 200 micrometer is your answer. Fine. So whatever rest over topic we'll cover next video. Till then revise, take care, bye bye.